Everyone, welcome to my library. And yesterday you must have studied about elastin, collagen, and keratin. Today, let's solve the questions, which are the most probable questions from this. Actually, I have, made you guys do the questions from quiz as well. So we'll have a plethora of some 10, 20, or 30 questions from these three topics, such that we are covering everything, which is, probably we are covering everything which can come in your exam. So let's start with these questions. First is 17th number question, which says an example of a structural protein which is also an enzyme is actin, myosin, trypsin, or troponin. You all guys know that we use an enzyme as trypsin in what? In digesting your protein first, and by utilizing the activity of its digestion, we utilize it in the protein sequencing as well. So therefore trypsin will be the correct answer. 18th question says most abundant protein in human body is, you have studied yesterday, in human body means extracellularly located protein is collagen, and after collagen comes what? Keratin or elastin. You can let me know, okay? So an example of a trimeric protein is isozyme, hemoglobin, keratin, or collagen. Always remember that whenever there is a term called trimeric, most of the answers will go in the direction of collagen, because you have trimers in the basic protofilament, basic protocollagen of this collagen fibrous protein. Then comes 20th question. Collagen is best described as first an alpha helical structural protein, B a coiled coil found in here, C a cross-linked globular protein, D triple helical fibrous protein. First of all, the answer would be triple helical fibrous protein. Now I'll let you know how can you eliminate the option. First one says alpha helical structural. So collagen is functional, not structural. Therefore, this option is incorrect. B a coiled coil found in here. It's not a hair protein, so again because of hair this one is wrong. And a cross-linked globular protein, again it's not a globular but it's a fibrous protein, therefore your option C is also incorrect. So correct option will be option number D. Moving on. So then we have a question as collagen is a fibrous protein consisting of three peptide chains entwined in a triple helix. What repetitive motif is responsible for collagen's ability to form the triple helix? Yesterday I have told you, आपको notes में it's clearly mentioned that you have what? You have glycine, X and Y. So where is it written? It's C, glycine, X and Y. So you can clearly remember that glycine is like 1/3 of the collagen is made up of glycine, proline, hydroxyproline, glycine, then proline, X in place of X you'll substitute it with proline, and Y can be hydroxyproline or hydroxylysine. Okay. Then 22. Which one of the following is unlikely to be a water soluble protein? So which is unlikely to be a water soluble protein? I've told you it's a universal thumb rule that your fibrous proteins are water insoluble, whereas your globular proteins have the probability that they can be hydrophobic, but in most of the cases they are hydrophilic. Okay, so the first option is ribonuclease. It's an enzyme. It has to be found, essentially, absolutely it has to be found inside the cell, therefore it will be a water soluble. Then myoglobin, no it can't be, because again inside the cell you require it. C is collagen, yes it can be because it's a fibrous protein. D is a lysozyme, which is again water soluble because it is essentially required in most of the conditions. So C will be the correct answer. 23rd is, which of the following is not correct about collagen triple helix? A, it's rich in glycine. It actually is rich in glycine. B is rich in proline. Yes, there is a lot of proline there because that gives it a rigid structure. C is rich in hydroxyproline. Again, hydroxylation is a characteristic feature of your collagen. D is an alpha helix. Definitely it is D, because see, after it becomes a trimer, it looks like a alpha helix. It forms a structure which is an alpha helix, but it is basically made up of what? Alpha chains. And alpha chains have how many amino acids? It has 1,000 amino acids. So it's basically very different from alpha helix, which is called as what? Alpha chains. Then comes 24th question. Choose the incorrect statement with regard to collagen molecule. A, it's formed from 3 alpha chain. Very well said, because I just told you that there are alpha chains. Then glycine accounts for 1/3 of amino acids found in collagen. Yes. C, it contains less hydroxyproline. This is debatable. D, ascorbate is essential for hydroxylation of proline. So basically you got three options which are correct. So it's asking incorrect statement. So first of all, it will be C. And for second reason, if you want to explore more, then elastin always has less hydroxyproline as compared to collagen. So collagen has a lot of hydroxylation on proline, whereas comparatively elastin does not contain that much hydroxylation. Okay, so 25th question is, which of the following statement are true about the structure of collagen? P says fundamental unit is tropocollagen. Yes. H bond between intra chain between three alpha chains hold the helix together. Yes, absolutely. Rich in proline and glycine. Yes. Three alpha chains wind around each other in a characteristic left-handed triple helix. That's what I just told you, that there are alpha chains which intertwine and form a helical structure. Chains intertwine to form a helical structure. So all the options are correct. It will be D, P, Q, R, S. Then we come to 26th question. Which of the following cofactors derived from amino acid is involved in the formation of hydroxyproline during collagen synthesis? So basically for hydroxylation, your enzyme requires a cofactor which is ascorbic acid or vitamin C. So it is asked cofactor derived from a vitamin. So basically ascorbic acid is vitamin C. So there is a cofactor which will be derived from vitamin C, which will be utilized by your prolyl hydroxylase for the hydroxylation of proline. Then comes 27th question. Collagen occurs in different forms which are usually described on the basis of A, type of carbohydrate present, B, cysteine content, C, hydroxyproline and hydroxylysine content, D, types of peptide chain present. See, collagen occurs in different forms means first you will synthesize it, then it will be in alpha chains from, then the alpha chains will combine together. See, alpha chains, and then alpha chains will combine together and will form what? Will form intramolecular hydrogen bond, and then they will form basically they have peptide bond present in them, and then when they will form what, they'll form dimers, then the peptide bonds are again linked with each other. So based on the types of peptide chains present, you can classify collagen. Okay, so basically you can also answer this question by this thing, that type of carbohydrate present, no carbohydrate. Where do you find the carbohydrate? At Olin glycosylation on hydroxylysine, right? So that can't be a characteristic feature because वो तो exception है. Then we come at cysteine content. No, you can't find that. See, hydroxyproline and hydroxylysine, which is ये variable नहीं होता hydroxyproline and hydroxylysine content, that you will find in every collagen because hydroxylation is very much essential. Okay, hydroxylation नहीं होगा तो cross linking में problem आएगी because L-lysine residues will not be formed, and once L-lysine will not be formed, then your aldol condensation will not take place and your chains will not link together. So then again the option which is left is D option. Then we come to 28th question. Choose the miss pair. Please read carefully that you have to choose the miss pair. So first is silk fibroin. See, here again, you know that collagen has glycine XY. So this is the miss pair because it is written phenylalanine, serine and proline. So out of this, you can write these informations in your copy or wherever you write your information, that silk fibroin has glycine, alanine, serine, elastin has glycine, alanine, valine, and alpha keratin has serine, glutamate and cysteine. Okay. So moving on. Which of the following is not a feature of alpha keratin? First is presence of sulfide, disulfide cross links between adjacent alpha helices. Okay, fine, very much probability because there will be cross linking. So your alpha helices will combine with the help of disulfide linkages. Second, presence of intrachain hydrogen bonding. Yes, it is absolutely possible because intrachain bond is always a hydrogen bond. Contains predominant amino acids like phenylalanine, cysteine and methionine. You just read that yes, it contains a lot of cysteine because of which it is able to form your disulfide, which is again methionine is there and phenylalanine is there. D, presence of intrachain SS crosslink. First of all, it's a very logical question that you can't have SS linkages within a single polypeptide chain. Like it can't cross-link on itself with the help of disulfide. Disulfides are mostly there to form multimers of protein. So intrachain is a problematic word here. Therefore, it's not a feature of alpha keratin. The 30th question says desmosine. Desmosine is a derivative of 4 lysine residues. A derivative of 4 histidine residues is found in the blood clotting protein. Thrombin is found in fibrous protein elastin. First of all, desmosine is the legitimate functional unit of your fibrous protein elastin. Elastin की जब सब unit आप बोलते हो तो you don't say that it's a topoelastin. You always say it's desmosine. It is a derivative of 4 lysine residues for sure. So the answer would be P and S. Also, if you eliminate the options, then you have correct option as S. Now you have to look forward for, you can cross these and you have to look for P and Q option. Okay. Q says it's a derivative of 4 histidine. No, histidine is not there. It's a derivative of 4 lysine residues. Whatever you have studied yesterday, as in collagen, elastin, keratin, you will get to see that there is a lot of glycine, there is a proline, there is lysine, but hydroxylation of lysine is deficient in certain of the your proteins. But essentially, आपको ये amino acids you will get to see that glycine, proline, lysine, and the hydroxylation on these amino acids. Then we come to the last question of this topic, that is disulfide bonds do not form in cytosol, but keratin intermediate filaments in skin are cross-linked by disulfide bond. This can be explained by the following. See, what happens is the question wants to say कि disulfide bonds जो है वो cytosol में नहीं बनते because your cytosol has a reducing environment and you know कि disulfide bond को अगर आप reducing environment में रखोगे तो it will become SH group. So the linkage will disrupt in the reducing environment. So it's asking लेकिन keratin filament in the skin are cross linked. केरेटिन में तो प्रेजेंट होता है, तो हाउ कैन यू एक्सप्लेन दिस फेनोमेना? ए सेज IF आर ट्रांसपोर्टेड टू द सेल एक्सटीरियर वाया द सेक्रेटरी पाथवे दैट अलाउज द फॉर्मेशन ऑफ डाइसल्फाइड बॉन्ड. नो, अगर सेक्रेटरी पाथवेज आपकी अलाउ करने लग गई डाइसल्फाइड बॉन्ड फॉर्मेशन को तो बहुत सारे लिंकेजेस फॉर्मेशन हो जाएंगी. एंड ऑल्सो आपकी अगर एक्स्ट्रा सेलुलरली आप ट्रांसपोर्ट करते हुए इसमें एंजाइमेटिक मॉडिफिकेशन कर रहे हो तो देयर विल बी एंजाइम जो कि इस मॉडिफिकेशन को रिमूव कर देंगे. तो ये फिजिबल पाथवे है ही नहीं, तो ये होता भी नहीं है. B. Your keratinocytes do not have a strongly reducing cytosol, thereby allowing the formation of disulfide bond in the cytosol. So yes, ये एक reason हो सकता है, but then again आगे के options explore करके I'll tell you the reasons. C. Keratinocytes lack glucose 6-phosphate dehydrogenase so that their cytosol is less reducing. No, it is not the case. D. Disulfide bonds in keratin are formed after cells have died and therefore cannot maintain the reducing environment in the absence of metabolic activity. So we were talking about this glucose 6-phosphate dehydrogenase which is in cytosol, which is utilized in your glycolysis process. But इस case में it's not the case क्योंकि keratin cells में जैसे ही keratins are formed, the cells are dead. cells की death हो जाती है. It's an adaptation just because of which इसका जो cytosol है उसमें metabolic activities are absent. And once your cytosol has no metabolic activity, that means उसके पास ग्लाइकोलिसिस की एक्टिविटी भी नहीं है. तो सी ऑप्शन इज इनकरेक्ट. डी ऑप्शन इज करेक्ट. अब बात करते हैं कि बी कैसे इनकरेक्ट है. बी बोल रहा है कि उनमें स्ट्रांगली रिड्यूसिंग साइटोसॉल नहीं होता इसलिए डाइसल्फाइड बॉन्ड्स बन जाते हैं. नो, बिकॉज उनमें साइटोसॉल ही लिविंग प्रेजेंट नहीं होता, बिकॉज इट डज नॉट कंटेन अ लिविंग साइटोसॉल, इट डज नॉट कंटेन द रिड्यूसिंग एंजाइम्स इन द साइटोसॉल बिकॉज द सेल्स आर डेड, राइट. देयरफॉर वो सिंपली वहाँ पे फॉर्मेशन हो सकती है क्योंकि अब वो रिड्यूसिंग एनवायरनमेंट नहीं प्रेजेंट है. ओके, सेलुलर कंटेंट्स का ऑक्सीडाइज होना मतलब डेथ होना भी होता है बिकॉज इनिशियली आपके प्रोटीन्स ऑक्सीडाइज हो जाते हैं एंड व्हेन एवर डेथ टेक्स प्लेस ये ऑक्सीडेशन का प्रोसेस बढ़ता रहता है. तो विद दिस क्वेश्चन थिंग कम्स टू एन एंड. आई होप आपने ये पढ़ा होगा एंड आपको कुछ समझ में आया होगा. थैंक यू एवरीबडी, कीप स्टडिंग. एंड इसके बाद हम स्टार्ट करेंगे शेपेरॉन से. आई होप आप पढ़ रहे होगे एंड आपका एग्ज़ाम क्योंकि बहुत जल्दी होने वाला है. सो थैंक यू एंड गुड लक.